Thank you very much for giving this excellent opportunity to share my work with you. It's an honor to be here. So I work on bacteria. I'm a bacteriologist by training and also a geneticist. So I particularly work on bacterial cell walls. I'm sure all of you know what bacteria are. Very small, ubiquitous in nature, present everywhere in deep seas and oceans and atmosphere and all that. And more so, they have gained more importance because now they are present in their vertebrate hosts, particularly in human beings. You can see that when human cells are something like 30 trillion, then the microbial cells are many more, and they're present all over the skin, gut, and mouth, etc. So they are unicellular and come up in different shapes and sizes. You can just see them here. But we work on a model organism called Escherichia coli, very well studied. People actually think that everything is known about E. coli, so why actually people study E. coli? But then we look at their cell walls, and then the question we asked several years ago was, I'm going to explain it now, what it is. So the bacteria are very small, though it's kind of drawn out of scale. It's something like 2 to 3 micron in length, right? And then this model organism, E. coli, is rod-shaped, right? And then, so if this is the cell wall, then it is covered by a sac-like molecule called peptidoglycan or cell wall. So I'm going to talk a lot about this peptidoglycan today. So it's something like a jute bag sitting tightly across the cell. So it needs to be elastic, porous, because molecules have to enter the cell and then go out of the cell. At the same time, it has to it should not leak out the materials which are present inside the cell. So it has it has to be very dynamic, and then at the same time maintain the shape of the cell so that it actually is elastic, right? And then the a property which it has is it's a single molecule. Okay, so what it means is it's a single large thing just sits across the cell. Now, then the question comes, when it is sitting like this, how is this? So whenever a bacterial cell grows, right, if you think this is a daughter bacterium, it has to grow into a mother cell to become big before it divides into two. This is called a very simple binary fission. And now this new bacterium again goes here and then grows and then divides again. And when there are favorable nutrients, this is how the bacteria grow and multiply very rapidly. When the nutrients are there, just this cycle happens in something like 20 minutes. So that is why the bacterial load is so much in everywhere, wherever you see. Now you can see that the gray colored line is the cell wall. That's how it is depicted. So naturally, the cell wall must grow when this daughter is growing to be a mother, right? Because the inside of the cell, which is called the cytoplasm, is growing, so the cell wall needs to grow. So why is studying the cell wall so important? Because from early on, the discovery of penicillin was that the penicillin inhibited the growth of bacterial cell wall, which means that the bacteria cannot become a mother cell, right? When you have penicillin here, this cell would lyse and then die. So there are several antibiotics which target the cell wall. They're very well known, the penicillins, the beta-lactam antibiotics, and vancomycin, etc. However, in recent years, there's a lot of talk about antimicrobial resistance, right? The antimicrobial resistance is very ancient, though people think that it has come out because of the human interventions. It is not so much so. Antimicrobial resistance is mostly because of the bacteria mutate so fast, right? Their evolvability is so high, so they always continue to have mutations which give rise to resistance to the antibiotics. For example, we are using beta-lactam antibiotics and the bacteria develop resistance to the beta-lactam antibiotics. Of course, the problem is enhanced and accentuated by the human use because we are overusing antibiotics, and then the antimicrobial resistant bacteria are getting enriched in these environments which have a lot of antibiotics. And then there is growing concern about that, more so because now we have superbugs. Superbugs would mean that the bacteria which have resistance to all the known antibiotics which we use. So the superbugs are increasing, and then the new discovery of antibiotics is decreasing. So since 2000, there are only 14 antibiotics which have been approved, and most of them are the derivatives of the old existing antibiotics. And then only 4 of them represent new class of antibiotics, and again, they are not very effective. So the need is there to identify new drug targets, right? So that we can make new antibiotics which target these new physiological pathways of the bacterial cells. So in this context, we thought that understanding the bacterial cell wall, how it grows, is of importance so that we can identify the new drug targets in the bacterial cell wall. Bacterial cell wall is a very attractive drug target because it doesn't give you toxicity, right? Because humans do not have any components of the peptidoglycan cell wall. So it is somewhat safer to use these antibiotics which target the bacterial cell wall. Right? So several years ago, we asked this question, then how does the peptidoglycan sacculus, which I described you just now, the so-called cell wall, enlarges during growth of a bacterium? So I had said before that this is the daughter cell, and this is a kind of, you think this is a mother cell. Now, how does this sac-like molecule enlarges in size? And I had told you this sac-like molecule is a single molecule, right? So what does that mean? So it is, chemically speaking, right, the white lines are the simply glycan strands, which are just made up of very simple sugars, okay? So now, and the white lines which are drawn horizontally here are the peptide chains, okay, which are made up of amino acids, which are linked to each other. In fact, those peptide chains are the ones which link the glycan strands and then make this molecule intact and then keep it close. Now, how can you now make this sac-like molecule enlarge in size, right, without disrupting the pore size and the overall structure? So which would actually mean that most likely the peptide chains are linked here, cross-linked here. So those cross-links need to be broken so that this new material, which is represented here in red, has to be inserted in the middle. So this is the question which we asked. Is this hydrolysis or the cleavage between these molecules, is it necessary for the expanding the cell wall? So if it is so true, then those enzymes which cut this will be required for the bacterial growth, right? So because I'm a geneticist, we took a genetic approach, did lots of genetic screens, and finally identified. Okay, so I had explained this, how the peptidoglycan, I'm so sorry. How the peptidoglycan structure looks like. So I had said that this is the glycan strand and this is the peptide chains which are cross-linked with each other. And we don't have to get into these strange amino acids. But then what I want to say is, now this is linked together by two kinds of bonds. Again, I mean, we don't have to look at these amino acids, but there are two different kinds of cross-links which which are there in the peptidoglycan cyculus. So using those genetic screens, what we identified was 4 enzymes, which we named them as MEP-SMH and MEP-K. MEP stands for murin endopeptidases. Murin is another term for peptidoglycan. Murus means wall in Latin, so people use the word called murin for the peptidoglycan. So what we identified was, now these 4 enzymes are absolutely important for the daughter cell to become the mother cell. So all these 4 cut the cross-links between these 2 glycan strands and make space, right, for the new strand to come in. This red arrow is the new peptidoglycan strand, which is made in the cytoplasm and going outside and then getting incorporated into the cell wall. And now that's how this, now 3 glycan strands would become 4 glycan strands, right? Now, we looked at the biology of these enzymes, identified the genes, and then studied them. So if these 4 enzymes are, so these are called as spacemaker hydrolases because they literally make space in the peptidoglycan for the incorporation of new material. And if these 4 enzymes are not there, so this is, these are all E. coli cells, which is our model organism. This is how a wild type E. coli cell look like, rod shaped. But if these cells do not have these cell wall hydrolases, so they do not grow in length, they become rounder, and then eventually then they die. So this shows that the hydrolases are important for growth, and then we have also shown that these are essential because they do not make the peptidoglycan cell wall and therefore lyse. Right. So I just want to give something like a trivial analogy to this, what I have explained. So if you actually think that this room is made up of bricks, right, and then you have to enlarge this room. You have to basically elongate this room. There are different ways of doing this, right? You break this wall, and then you can extend this. You can break that wall, extend it, or you can do both, right? Extend both ways. Or the other way is you actually remove the bricks in the middle, right, in the body, and then put new bricks. And bacteria enlarge their cell walls in all 4 different ways I have told. But most model organisms, like E. coli, what I use, take the last alternative. That is, it breaks the wall in the middle, right, and then put the new bricks. So that's what is kind of represented here. And what I kind of think is, if these are the bricks here, now, right, now you need to remove something like the cement in between the bricks, and then there's space. Put the new bricks, which are the peptidoglycan precursors, and again seal them off. The sealing enzymes are the synthases, which are the targets of beta-lactam antibiotics and which are extremely well studied. Right? So now this represents the activity of these hydrolases or enzymes, which I just told you. So they make space in the middle so that the synthases come and then they fill up with the new material, and that's how it would elongate. So in layman's terms, what my lab has figured out is something like the laborers who come and break the wall, right? But then the question comes, who tells the laborers to break the walls, right? They just cannot come and then break the wall anytime whenever they want. So the laborers need to be told something like a supervisor, right? That this is the time to break the walls, right? So now we also figured out, so the supervisors again would mean that the proteins which regulate these hydrolases, right? So very often these hydrolases are repressed, right, or kept in off position so that they do not cut these walls randomly. So they cut these walls only when there are nutrients around, right? And whenever there's a need for the peptidoglycan or the cell to enlarge, that is the time the hydrolases would act. And then there are proteins which do that job, right? Again, when I say a supervisor knows or tells the hydrolases to cut, again, who tells the supervisor, right? What is the signal that the supervisor gets to say that the room needs to be enlarged? That is where comes almost like the bricks and the cement are the peptidoglycan precursors or the nutrients which indirectly tell the supervisor, now the material is there for the cell to enlarge, so that then the hydrolases are stabilized, they cleave the crosslinks, and then the new material comes, and then the cell would enlarge. So to summarize now, so what we have shown is the peptidoglycan hydrolysis or breaking the cell wall is extremely fundamental to make the cell wall, right? So initially we had shown in E. coli, last 5 or 6 years since we have shown, now the same process has been shown in several bacteria, including both gram-negative and gram-positive ones, like Bacillus, Vibrio, and Pseudomonas, and Streptococcus. And what is important here is, what we figured out is the breaking the wall is the committed step for the cell wall synthesis, which means once it breaks, there's no turning back, right? Once the breakage happens, the synthesis has to happen. So that's the rate limiting or the committed step. That is why that process is regulated very stringently, so that they just don't act and then break the wall. Right? And then inhibiting this process is a good alternative strategy to kill bacteria. So the enzymes which we have identified here, the breaking enzymes, one particular class is something called MEP-S family, which has no known inhibitors. So we are actually trying to look for the inhibitors for those enzymes. So that's the future plan. And now this is my present lab at CCMB. Unfortunately, I didn't have the picture of my past lab mates who do this. And thanks for your attention.
Thank you so much, Professor Reddy, Dr. Reddy. Sorry.
From the images, from the sort of cartoons you drew of the cell wall, it looked like it had a random structure. Is it a regular array of the peptide and glycan bits, or is it random? Because the growth mechanism would be very different for the two cases.
Yes, I do not think it's random, but people don't know the precise structure because only the cryo-EM the pictures can tell you whether the peptide chains are arranged randomly or regularly, but people do believe that it's quite random.
That it's quite random, is it?
Yes.
I see.
I think there was one more question at the back. Can you just hand the mic to one of the students there?
Hello. I wanted to know, how does these hydrolases distinguish between new strands of peptidoglycan which is being incorporated and the ones which are already being there? So it has to know which is already being there and then break the strands and incorporate new. So how does that distinction happen?
So most probably that distinction, they do not do that distinction, right? So that is why they're so kind of very stringently repressed all the time. So the action of them would open up, right? And the new strands come. When the new strands are coming, again the hydrolase activity is repressed. Right? So they work only at one particular window of the time, and that is pretty critical.
Okay. And one more question. Is the insertion of these new strands, is that random, or there is a particular positions in a cell where it will be incorporated?
So people think that it happens in the body of the cell, random and then diffuse, and then it is coupled to the cytoskeletal movement in E. coli, which is MRE-BCD. So they are the ones which determine where the synthesis is happening. But we still don't know the connection between MRE-BCD and these hydrolases. That's something to work out.
Thank you.
Thank you so much, Dr. Reddy.